Hi, Bio 30. I'm going to do a quick lesson today, continuing from mitosis. I'm going to post a lesson on meiosis today. That's slides 27 to 38 in your notes. There's a good, just like with mitosis, there's a really good diagram on page 564 in your McGraw-Hill, looking at the stages of meiosis. Okay, so make sure you take a look at that. This whole discussion about homologous chromosomes and sister chromatids is going to be a really big deal in this lesson today. So remember, mitosis is the purpose of mitosis is growth, development, and metabolism. The purpose of meiosis is to produce haploid cells rather than diploid cells, right? So we end up with cells at the end of meiosis that have 23 chromosomes rather than 46. So the cells will be N rather than 2N. So there's two main things that happen in meiosis that really differentiates mitosis from meiosis. The first is reduction division. So we produce daughter cells with fewer chromosomes than the parent cells. The next one is recombination. So in meiosis, rather than producing clones of the parents, the daughter cells are all genetically distinct. So there's recombination, so there's jumbling up of alleles that happens. And we make genetically distinct gametes. And that's a way that we increase genetic diversity. Okay? There's also two stages in meiosis, meiosis 1 and meiosis 2. So meiosis 1 is where all the different stuff happens in comparison to mitosis. Meiosis 2 proceeds much like mitosis, but meiosis 2 has haploid cells rather than diploid cells. All right? So when we look at meiosis, remember, so think, when you think meiosis, not mitosis, when you think meiosis, think IPMAT, PMAT. So this would be meiosis 1, and this would be meiosis 2. Okay? Now all the different stuff happens here. All right? So this would be interphase. So interphase proceeds the same way. We're going to replicate the DNA in the S phase of interphase. And then you're going to have actually prophase 1, metaphase 1, anaphase 1, and telophase 1. Telophase 1 may or may not happen. Then you have prophase 2, metaphase 2, anaphase 2, and telophase. All right, so we're going to produce 4 genetically distinct gametes at the end of meiosis, whereas we only produce 2 clones of the parents in mitosis. All right, so in your notes here, let's look at the stages. So this is very different. So prophase 1, okay, you have this organization of cells where the homologous chromosomes actually line up side by side. That does not happen in mitosis, and this is called a tetrad. So I'm just going to draw this up here. So if I look at prophase 1 and I look at prophase of mitosis, here's what happens. So you get homologous chromosomes lining up side by side. So the same thing happens in mitosis. Spindle fibers start to show up and centrioles start to show up. The nuclear envelope dissolves and all that stuff. But in mitosis, the homologous pairs are like randomly assorted. They line up side by side in prophase 1. And this is called a tetrad. And it's called a tetrad because there are 4 pieces of genetic information when the homologous pairs line up side by side. Okay? So you have this tetrad, and then the 2 homologs have the sister chromatids. So this is also a big one here. So if I look at meiosis, right? Again, this is a homologous pair. Okay. And then the homologous pairs each have their duplicates, and those are called sister chromatids, right? Okay, so those are called sister chromatids, but you also have sister chromatids across the homologous pair, right? So you actually have sets of what we call non-sister chromatids. Where the chromatids of each separate homolog are also associated. So the deal is with prophase 1, you have this tetrad, and then this thing called synapsis happens. We also call this crossing over. Okay. So if I look at my notes, this is synapsis. So the diagram here has the homologous pairs shown with different colors. But what they do is they swap pieces of genetic information. So if I have, I'm going to take this homologous pair and then I'm going to color one red and one orange and then bring that down here. Okay, so that's the homologous pair here. What they're going to do is, in meiosis, these guys are going to exchange information across the homologous pair. So you're going to have jumbling up of maternal and paternal chromosomes. And this is a way that we increase genetic diversity. So alleles for the same traits get exchanged when this happens. And we end up with some unique gene combinations. Right? So this is a way to increase genetic diversity. Okay. So we get these pieces of genetic information being exchanged during prophase 1. That does not happen in mitosis, right? So this is mitosis. Okay. This exchange of alleles does not happen. So we have recombination going on between non-sister chromatids in prophase 1 of meiosis. Okay, and this can happen at multiple spots. Like their exchanges could happen in multiple places, not just one, across the homologous pair. Okay, so the equator here is right. So the equator here is this way rather than this way. So metaphase 1, metaphase in mitosis, if you remember, right? What happens is the homologous chromosomes line up along the center of the equator in single file, right? And then you have your spindle fibers and your centrioles, the borders. So that's mitosis. But in meiosis 1, what actually happens here is, so here's the equator. And I'm going to use the different colors again. The homologous pairs do not line up single file. What they do is they line up side by side. Okay, so the homologous pairs line up side by side rather than single file. That is unique to meiosis. Okay. So the spindle fibers do the same thing. They'll attach to the centromere. And so here's the big difference now. With anaphase 1, okay, what happens with anaphase 1? If I draw this again, is that one homologous pair gets pulled this way of a set. Okay, and then one homologous pair gets pulled this way. Okay, so the homologous pairs separate. And that is not what happens in anaphase of mitosis. Right? In anaphase, right, if this is a single homologous pair, they'll get split. And so this is maternal and this is paternal, right? There'll be a paternal chromosome that goes this way and a maternal chromosome that goes that way. And then this way, same thing will happen, right? Paternal and maternal chromosome. So you get both sets of homologous pairs that you need in anaphase of mitosis. But here, in anaphase 1, okay, you get paternal and maternal. That would be maternal and paternal, right? So these are two sets of homologous chromosomes. You either get one homolog or the other. So you get a homologous chromosome with a sister chromatid. You don't get both sets of homologous chromosomes moving away from each other. And then this will end up, this will end up in tubulophase 1, and then we'll end up with two cells that either have one or the other homolog, not both sets. Right, so we won't end up with both homologous pairs. So that would be diploid. And then these are now haploid cells. And they're haploid because you don't have the full set of homologous chromosomes. You have one homolog with a sister chromatid. Okay? So that is fundamental to understanding the difference between mitosis and meiosis. Okay? All right, and then so telophase 1 may happen or may not. It might just progress into the second stage of meiosis. But either way, when the cell splits at the end of meiosis 1, you're dealing with a haploid cell. You're not dealing with a diploid cell. And then meiosis 2 proceeds the exact same way as mitosis. So you'll get, in anaphase, you'll get the chromosomes lining up along the equator, and then the sister chromatids split. But the cells are haploids, so that's the big difference there. The cells are haploid, so you get haploid gametes. Okay? So let's summarize this. So main differences between meiosis and mitosis. And I'm going to give you just some visuals on the board here. So synapses for crossing over. Metaphase 1, homologs line up on either side of the equator, not at the center. In anaphase 1, the centromeres don't split, but the homologous pairs do. Sister chromatids remain intact, so you get one homologous chromosome going to one side of the poles of the cell, and the other homologous chromosome going to the other pole, and those will end up in different cells. You don't get both homologous chromosomes. Meiosis 1 produces 2 haploid cells. Meiosis 2 produces 4. Mitosis results in the creation of daughter cells that are precise copies of the parent. And the outcome of meiosis is genetically distinct gametes. Okay, with recombination, you have two things. I'm going to talk about this later with Mendel, but you have the law of segregation, which tells us that alleles of this same trait assort without being linked. But you also have independent assortment, where alleles or genes of different traits, so like hair color and eye color, for example, they also differentiate during gamete formation without being linked. Okay, so So segregation tells us that alleles of the same trait are not linked. In independent assortment, the law of independent assortment tells us that genes of different traits are also not linked when gamete assignment happens and crossing over occurs. Okay, so let's visualize this a little bit more. Just give me one sec. Okay, so I just cleaned my board off. If we look at mitosis and just how mitosis proceeds. Okay, so remember that you have a diploid cell, and then S phase happens. And then you still have a diploid cell, but now you have a diploid cell with homologous pairs that have sister chromatids. And then mitosis happens. Right, so mitosis happens, and then you end up with diploid daughter cells. Right, so that's mitosis. Now if I look at meiosis, my board's a little wet here still, but if I look at meiosis, here's what happens. So it starts the same way. Right, you have a diploid cell, and this is in the germline of the gametes. Okay, so if we go back to reproduction, meiosis is happening in the Sertoli cells. Right, so the Sertoli cells have germline stem cells that turn into gametes, and then you have cells that nourish and help support the developing stem cells that will turn into sperm. Okay? So you have this, right, and then you have the S phase of interphase. So this starts the same way, right? All right, so cells duplicate, so that's now diploid. And then you're going to have your first division, and that's meiosis 1. Okay, so what happens is this guy goes here, and this one goes here. So you end up with a single homologous chromosome with a sister chromatid intact still. All right? Now if I were to draw that another way, right, let's draw it like this. Okay? So we have that happening, okay? So you'll get this in either cell. Okay, that'll end up in a cell. And that'll end up in another cell. And in fact, if I want to be more exact, what I will do is I will show what I'll do is I'll show this, right? The homologous pair is different colors, and I'll show the crossing over. Okay? Right, so we'll end up with, this sort of deal happening. Okay? So you'll have this. So here's the deal, is now this cell is haploid, but I'm going to draw it like this. So I'm going to draw it like this, n squared. And the reason why I do that is because it's a haploid cell, but it has an extra set of genes. Okay, so it has the sister chromatid attached to it. So it's two pieces of the same genetic information with your crossing over that's happened, right? So some alleles swapped, but it is still, right? It is still haploid because there's not two sets of homologs. So there's, so there's only maternal or paternal chromosomes. There's not both. Okay, then second division happens, and this would be meiosis 2. Okay, second division happens, and this would be meiosis 2, and then you end up with haploid gametes, and these are all haploid. And that will make your sperm and egg cells. All right, so that's what happens with meiosis. Now, I'm going to show this process as a graph, okay? So this should help visualize this as well. So if I draw a little graph here, and we look at this as the progress of meiosis, let's say progress of meiotic division. Okay, so this is the progress of meiotic division, and then this is the time, and then this is, let's say this is the amount of genetic information on my y-axis. This is a question on the mitosis, meiosis, sexual, asexual reproduction exam, by the way. And let's say I've got, so this is just like your gross amount of genetic info, and we've got haploid, diploid, and then we've got genetic information doubled. So we don't actually have tetraploid in meiosis, but you have, if I visualize this, that's this, right, and then, right, so that's this potentially, or this, and then that would be this. Okay, so this is homologous pairs with sister chromatids. That's a homologous pair, and that's a single homolog with a sister chromatid, and this is a haploid cell. Okay, so if you think about what happens in meiosis, we start with, right, a normal diploid cell, and the diploid cell is just a homologous pair. Then the next thing that happens is we replicate our DNA. So the amount of genetic information is actually doubled, right? So we go from 2N to 4N, but it doesn't function as 4N. It functions as 2N still, right? So we go from here to here. Okay? So this is S phase. Now, the next big phase that happens is meiosis 1. And think about what happens with meiosis 1 is we end up like this. Right? So you end up with a single homolog, but you have a homologous chromosome with a duplicate. So it's actually the same amount of genetic information as a diploid cell, but it's half the genetic information doubled. Half the required genetic information that you need, but there's an extra copy of that half. Okay? So this would be meiosis 1. Okay? And then we go through meiosis 2, and we split the homologs. So you end up eventually like this. Okay, and then this is your graph, amount of genetic info over time. And that's what it looks like. Okay, so you can visualize that, then you've understood meiosis. And I'm going to leave it there today. Not a super long lecture, but you have mitosis and meiosis complete. I guess the other thing I'll address is your meiosis. So you can do the meiosis and the mitosis quiz now. But I have posted on D2L a meiosis assignment, and I'm going to show that to you. Now, if we were in class, you would do meiosis as an interpretive dance, but we are not in class. So, you're going to draw me a comic strip with meiosis. So all the criteria is up. Please read it. Okay, you need to include all these steps in meiosis. So the purpose of meiosis, how the ploidy changes, what reduction division looks like, crossing over synapse, homologous chromosomes. Include the stages and phases of meiosis. Happens in gonads, right? Number of cells produced. Now, one thing where you should watch the video, the meiosis square dance. It's an old Discovery Channel YouTube video. Okay, so watch the meiosis square dance, and that'll help summarize it. But what I want you to do is I want you to come up with a creative comic strip, and I want you to think of some sort of comic book scenario with frames that will help serve as an analogy for the stages of meiosis. So, in the past, people have done like SpongeBob SquarePants or like some sort of alien replicating itself, and the Avengers have to fight it off or something like that. It's up to you. But either way, you have to use frames in a comic strip as an analogy for meiosis. And what you need to do is do that. And then explain how each frame represents meiosis and what phase of meiosis it represents. So whether you do that on the back of your comic strip or you provide a separate sheet of paper with it, I want a picture of the comic strip, and then I want explanations of each frame. So like frame one, frame one would sound like something like this. SpongeBob SquarePants is doing this or that or whatever. Replicates his DNA to make more SpongeBobs. This represents this phase of meiosis because, and then create a story from that, and a conclusion, and use the different phases of meiosis to help guide you create your comic strip. All right. So I'll put a due date up on this later, but you can do your meiosis comic strip in pairs. Okay, so you can do it in a group of 2 if you would like, and I'll put a due date up on the news feed. So that's all I'm going to talk about today. I will likely add another lesson tomorrow or early next week on different types of reproductive strategies, and after that we'll be on to Mendel. So if you have any questions, let me know. We'll talk to you later. Bye.